chapter 25, urinary system. As you know, urinary system involves the kidneys, the ureter, urinary bladder, and the urethra. So it's a fascinating topic, and all these components, you had the idea of it, what they look like, where is the location in anatomy. So here's the kidneys, all right? Blood goes from the renal arteries towards the kidneys. After it branches out to several different types of arteries and arterioles. Uh, the blood is going to go to transfer to the tissues of the kidneys, and there are tiny little tubes inside of the kidneys. Those are called nephrons, and blood gets filtered there, and the waste and the excess material exit. All right, and as they exiting through the ureter, and the ureter linking the kidneys to the urinary bladder. So the urinary bladder is a temporary storage for the urine, right? And finally, urethra, which again you learn about the internal and external sphincter, and it allows the empty or uh, pretty much the exiting of the urine will take place, which a lot of times this is referred to as urine output. Okay, what amount is going to come out is basically referred to as output, urine output. All right. So there is a lot of nitrogenous waste, all right, urea, and uh, excessive waste, substances that we don't need or we have it too much. You know, the idea of the kidney is to balance things, and that's where at least majority of the function comes in. So let's go ahead and take a look at the function. It can balance the water. You all have that ideas already. In other words, when you are uh, drinking a lot of water, you're using a restroom more often, your urine output is higher. When you drinking less, obviously your urine output is going to be less and your urine will be more concentrated. So solute concentration, these are mostly referred to the ions. We have a lot of ions in our diet. They get absorbed in our small intestine going into the blood, right? But we may not need so many different types of ions. But these ions need to be balanced. Say, for example, that you're drinking or in your meal, there is a lot of sodium chloride. Sodium chloride is for salt, all right? So it's a solute, all right? Now, what is going to happen is, as we mentioned, these are source of a solute in our blood. So in our blood, now you have a lot of Na plus and Cl minuses, and this could be excessive because these are solute now. And in the capillaries, as you know, when we were discussing the cardiovascular system, we said the capillaries are thin, and it allows some of those water to come in. So your tissues or your cells will lose the water. The water is going to go more towards the solute, and it's going to increase the volume, and as a result of that, increase the blood pressure. All right. So obviously, we don't want that to happen, even though it is happened. Somehow we got to get rid of these sodium and chloride and such. So again, the urinary system, if we can eliminate these, then there will be less of these available. Then there won't be, as much water reabsorption, And as a result of that, everything is going to be balanced. So the idea is the balance, as you can see, and the kidneys doing a good job of that if it's working appropriately and is healthy. However, your urine in this matter would contain more salt. So your urine will be more concentrated, more salty. All right. Regulating the extracellular fluid ion concentration. So again, same ideas. All right. Extracellular fluid is the fluid outside of the cell. All right. Ensure long-term acid and base balances. Our body actually works better to deal with the acids, and I told you this before, rather than the base, but they could do both. So I'm just going to give you an example. If you have excessive hydrogen ion due to having too much acidic food in your diet, and this acid is going to absorb in a small intestine, going to the blood. Now you have excessive amount of hydrogen ion. Again, the kidneys will help to get rid of this hydrogen ion. Now your urine is going to be acidic. Urine, for the most part, is semi-acidic. The pH of the urine is somewhere around about 5 to 6, which again, these are the pH scale. So it's a semi-acidic. Why is it so? It's because it's getting rid of this hydrogen ion. That's why. All right. So in a way, once you get rid of it, then you don't have to deal with having acidosis in your blood because you got rid of it. Well, does that mean we're never going to get acidosis? No, that does not mean that. Sometimes the hydrogen ion is way, way too much, even for the kidney to handle that. All right. And sometimes the kidneys are not working properly. So all of those could be problematic, obviously. Metabolic waste, as I told you, urea and the nitrogen waste and toxins and drugs, right? A lot of times drugs remain in our body for a long period of time. uh, as your liver working and other organs to doing the detoxification, slowly you're breaking things down, and now the byproducts needs to be taken care of. Some of those drugs and toxins can be exited, and sometimes it might take months in order to do that. All right. Therefore, the drug can stay within the system for long period of time. Depends on the type of a drug, by the way. But if a person is drug addict or perhaps using some kind of a drug, it is going to be in your system. So good luck if you are applying for a government job that they take in your urine sample and they test you for them, because it will show. All right. Um, Again, some drugs can stay within the system at least up to 6 months. All right. Same thing with the athletes. Sometimes, they use illegal drugs, anabolic steroids, things in that nature to have a competitive edge. But when you take a sample of the urine, all right, those byproducts as well as some of those drugs end up in the urine. And now you have to answer the question, what is it doing there? All right. As you know, a lot of athletes got caught that way as well. So there you go. These are the general function. Uh, Other type of function is the endocrine function. As you know, the erythropoietin or the EPO, we discussed this already. They are triggering the bone marrow to release more red blood cells, especially when your red blood cells is lacking or you need oxygen. We went over this quite a bit in a previous lecture, so look at it. And we mentioned that the EPO is generating mostly by the kidneys. There's another endocrine function, and that is called renin. And today we're going to be looking at how it can regulate the blood pressure. I told you that not all the blood pressure was covered during the cardiovascular system because many of these were related to the kidneys. And if you looked at some of the questions that you were not able to answer, hopefully today you could go back and answer them as well. Activation of vitamin D, that's also another thing. As you know, vitamin D is important in bone synthesis, so it can activate that as well. All right. So we went kind of like over these organs already, where they are and what they do, and that was more like anatomy. I showed to you guys which one they were what and so on. Interestingly enough, earlier I told you that one of the things that contains the urine is hydrogen ion. So if you have a hydrogen ion because you're trying to balance the acid in the blood, all right, and it end up in here, and I told you the pH is about 5 to 6 for the most part, all right. Uh, these acidic residues now prevent a lot of microbes going in, right? Urine considered to be pretty sterile, right? Because of having that acidic substances in it. So again, it's a way to prevent microorganisms to get in. Not only you're balancing your acid in the blood, but also you're preventing the microorganism to get in, because microorganisms usually they don't do very well in acidic condition. All right, so you have kind of like a natural way to protect yourself. All right, and microbes a lot of time have a hard time to get through your skin and integumentary system. They like to find other ways, including your respiratory tract, your oral cavity, and now we're talking about the urethra. All right. As far as the oral cavity goes, most of the time that ends up in the stomach, and your stomach is acidic. That's being taken care of. As far as the respiratory, that's a very delicate spot for the microorganism to get. But if you have mucus lining and so on, you have some protection. But again, it's not protection throughout. All microorganisms, as you know, influenza or common cold, and now coronavirus is being transmitted mostly using the respiratory tract, and all kinds of respiratory tract infection. But another way to get in is this urethra. All right. So that's another thing that you wanted to think about. Um, Internal anatomy of the kidneys is important because we're going to be talking about these structures. The renal cortex is towards the superficial region, and the renal medulla is more deeper region. Again, medullary pyramids, if you recall, was what we discussed. So here are the renal cortex. So I'm just going to highlight it for you so you'll see the cortex area right over here. All of these are the cortex areas, all right? And triangular shape areas, those are the medulla, all right? So medullary pyramid we discussed. So renal medulla or medullary pyramid would be, in this case, the blue region. All right. So what you wanted to understand is, if we have, let's say, a blue region right over here, say, for example, this is the same thing as here. And then on top, we had the cortex right over here. That would be the red region, this part, okay? So there are tiny nephrons in there that we're going to get into, and hopefully you remember different parts, all right? So there are tiny little tubes everywhere, and you could have so many of them, and so on. So these tubes ending up right over here. In fact, if you look, I want to erase few of these so you could see it a little bit better. You see these lines right here. These lines are these tubes right there. Okay, so these tubes are being empty in these spots, minor calyx and major calyx and renal pelvis, and then empty into the ureter. So let's say you have a lot of waters, all right, the water in the blood, it gets filtered, and then again, it goes into these tubes, and then end up in these regions and end up in the urinary bladder ultimately from the ureter, and then your urine output is going to be higher. Okay, so these tubes, as you know, by the way, I expect you guys to know. I'm just giving you a review right now. I expect you guys to know all the parts and the details of these parts as well. So again, in anatomy, you should have covered that. All right. So a student, even in this semester, always asks me, "Oh, well, we didn't cover that in anatomy. Or, well, we didn't get into, we didn't have time, and this and that." So now you understand. Now you know that you're behind. Now you know that probably your instructor didn't do a good job going over things. All right. Here is the cortex. The bottom line is the medulla on the blue. But what I want you guys to know is as you go down towards the medulla, all right, osmolarity increases. So this part you have higher osmo, the more up you have lower osmo. We'll discuss this later, but right now this is very important, all right, to understand about the osmolarity. Okay, so next up, I would like to go over perhaps different part of the nephron. So each uh, nephron are those tiny little tubes. So basic, functional, structural unit of the kidneys are nephrons. All right. So again, they filter the blood, they do all those things, they get rid of the waste, they get rid of the excessive stuff, all through these nephrons. So nephrons are important. In a single kidney, we have about 1 million nephrons. All right. But we're just looking at one. There are generally two types of nephrons. All right. 85% of the nephrons are known as cortical nephron, and about 15% of the nephron are known as juxtamedullary nephron. So if we want to know the difference between the two, again, in anatomy you discussed, here is the good example of cortical nephron, and here is a good example of juxtamedullary nephron. Here is the juxtamedullary nephron. Juxta means next to, medullary means medulla, all right, and it's a nephron. So remember, the top part was the cortex, and the bottom part was the medulla. And one of the important arteries separating the two parts together was the arcuate artery. In anatomy, you learn that, which is right here. Either way, what's the difference? Well, 85%, like I said before, are cortical nephron, all right, about 85% of the nephrons. So we're going to put our concentration here, and about 15% is the juxtamedullary nephron. Juxtamedullary nephron, the loop of Henle is much deeper, goes into the medulla, versus the loop of Henle or nephron loop. This is a loop of Henle or nephron loop. All right, is shorter. It doesn't go extended that much in the medulla. Majority of the parts happen to be on the cortex area, as you can see. Okay, now the juxtamedullary nephron deals with making concentrated urine. All right, right now we're gonna put our concentration on the cortical nephron. Okay, which again it does a lot of different things, and that's what we're going to go ahead and take a look at. All right, so you can see a lot of capillaries around them, And these capillaries are known as peritubular capillaries. All right. And major blood vessels going in is afferent arteriole. All right. And the blood vessels coming out is called efferent arteriole. And these ball of capillaries were labeled and known as glomerulus. All right. And the structures around them is called the Bowman capsule, or also known as glomerular capsule. All right. So I will refer it to both in my lecture so you could see it. And bottom line is, blood is going to enter in here. It gets filtered, it gets processed, and so on. And then again, at the end, right over here, those are where the empty into the tip of the medullary pyramid. Those were the waste and the excessive stuff and all that stuff that we discussed. All right, and we will be discussing. All right. Now, the blood vessels associated with the juxtamedullary nephron is just called vasa recta. That's all. All right. So, for a reference point, capillaries around the cortical nephron are peritubular capillaries. So that's something that we wanted to talk about. All right. So what we're going to be doing, we're going to dive inside of a nephron, right? And we're going to see what is happening. If we understand what happens in one nephron, maybe we'll have a better picture of what's going on. Okay, so let me go ahead and give you an idea about the nephrons and the different parts of it, and then we'll break it down. We'll look at one part at a time, and we'll see what we could do. Okay, so let's see. Um, we can do this. Let me see if I can go to this page. All right, so we could do this together. Uh, See if you can name this structure, so that way I don't have to write everything down. This was the Bowman capsule or glomerular capsule. All right, and then these were kind of like convoluted, was proximal convoluted tubule. All right, and then you had the nephron loop, which was loop of Henle. The loop of Henle that is going downwards was the descending loop, and as they're going upwards, that was the ascending loop. And then the last part of the nephron was DCT, all right, distal convoluted tubule. And then again, the very portion that extends all the way towards the bottom of the medullary pyramid was a collecting duct. So this was collecting duct. This one was labeled as distal convoluted tubule. This one is loop of Henle, also nephron loop. Again, this part is descending loop, and this part is ascending loop. This one was proximal convoluted tubule. So remember, this one is D. This one is P. All right. And this part right over here we mentioned is Bowman's capsule, also known as glomerular capsule. Blood vessels that come into here is the afferent arteriole. And the efferent arteriole is a little bit smaller in diameter. Efferent arteriole, if you recall, there was a smaller diameter. And This one right over here was the glomerular or glomerulus capillaries, if you recall. Glomerulus, all right. Uh, and then after that is all peritubular capillaries. So this is going to go everywhere, kind of, you know, and these would be the peritubular capillary. Okay. So I should say capillaries. All right. So because there are many, right? What we're going to do is first we're going to go ahead and take a look at this region right over here. Um, I'm going to highlight it so we can see it a little bit better. All right. So right now we're going to go ahead and take a look at this region right here. Right. So in order to take a look at this region first, we must see what is happening and what do we have in our blood. If we analyze the blood, right, and take a look at what we have in our blood, okay, here's what we can see. Um, so let me just extend this. This is from the renal arteries, right? And now we need to find out what is in our blood. So uh, you could think about this. We have water in our blood, we have waste in our blood, there are proteins in our blood, we have amino acids in our blood, we have glucose in our blood, we have different ions in our blood, um, we have bicarbonate HCO3 minus in our blood, uh, we have what else? Proteins, amino acids, glucose, different types of ions, waste, water. We do have other stuff like we're not going to deal with right now because of the fact that, we don't do much as far as the kidneys for it. So, for example, you could have cholesterol in your blood. All right. So those things uh, we really not regulating, and we don't care too much for it at this point in time. And I think we should be okay. But those are the majority of it. And we also have, don't forget, we have RBCs, red blood cells, we have white blood cells, we have platelets. Using our cardiovascular knowledge, you already know those. So think about what was on the plasma. Think about when you spun the blood, what did you get? And you come into a contact of it. Now again, there are hormones in our blood. There are other things in our blood, but right now we're going to concentrate on these. So all of these material, due to the force of the heart. Remember, the heart is contract and the blood is going to reach via the systemic circulation towards the different organs, including the kidneys. So ultimately, renal arteries, and after that, so many different type of arteries that I'm not going to get into at this point in time. You're learning anatomy, but ultimately, it's going to go to afferent arteriole. Afferent means arriving, so all this stuff is arriving. Efferent exiting, uh, they are exiting. But once you get to the glomerulus, you know, a smaller stuff they can pass through. All right, larger stuff they cannot. So this is where the process A takes place. Uh, Process A we can put as filtration. Filtration. It happens to be right on the vicinity of the glomerulus and the Bowman's capsule. Now we need to see how and what material can pass, one material cannot pass. Okay, so this is based on size only. So all the large material, they do not pass. At least we're talking about the functional kidneys, right? So platelets, they don't pass. White blood cells, they don't pass. Red blood cells, they don't pass. They're already too large. I mean, you can think about the disaster if they were passing. If they were passing and end up in the urine right here, all right, your urine is going to be red because you're losing some red blood cells, and you know the aftermath of it. I mean, your oxygen carrying capacity would be lower. You lose the white blood cells now. The white blood cells is even larger than red blood cells. So when you lose that, you lose your immunity. You lose the cells that's supposed to serve and protect you. When you lose platelets, you will not be able to, do the blood clotting. So those are too large to be passing. Proteins. Now, there are some proteins that are small, but majority of the proteins are too large. All right, they might play important role. Hemoglobin, enzymes, antibodies in our blood, those type of things. For the majority, a lot of larger protein they cannot pass. All right. If they cannot pass, what happens? They remain in our blood, doing their job, doing whatever they're supposed to be doing. By the way, proteins as well as ions, they can maintain osmolarity. That was a very important concept we learned on chapter 3. All right, we further discussed this on the cardiovascular system as well. So the amount of water goes in and out and so on depends on the osmolarity. If the osmolarity is too high due to excessive protein, more water can go in. All right. If it's too low, more water stays out. You get edema and all that stuff that we discussed, right? So it's important to balance things. Everything needs to be balanced, and it's all about the homeostasis. All right. So now what? Now this stuff is passing. All right, or the ones that I'm putting a check mark next to them, those are passing, right? So the ones that are passing is ending up on the inside of the nephron. And those is stuff, the check mark, we call filtrate. Filtrate are the material that are passing. If you brew your coffee, for example, and use a filter paper, the water and tiny little coffee granules as they get dissolved, all right, and end up in your coffee mug, that resembles and known as filtrate. That's what it is, all right. So what causes this filtration? It's worth taking a looking at it again, all right, and dive in and learn more about it. Let's go ahead and take a look at that. That is a little bit of anatomy involved. And for those of you that wanted to review this part, you kind of like on your own a little bit, but I'll go over the basics so you understand and you could read a little bit on your own as well. All right, so let's go ahead and take a look. Here's the afferent arteriole, here's the efferent arteriole, here's the glomerulus. All right, when it comes to the glomerulus and when we enhance or enlarge this part, we could see that these are not any capillaries right here. All right, these are not any ordinary capillaries. Glomerulus is a special capillaries that is fenestration, has a fenestration or tiny little pores in there. All right. Normally, we mentioned that the blood pressure in the capillaries they're about 20 to 40, all right, millimeter Hg. These capillaries, the pressure is about 55. Again, they have tiny little holes, and they're specialized in doing the filtration and such. so that way they can hold up more pressure. All right. On the other hand, the afferent arteriole is wide as it comes to the efferent arteriole. Look at the diameter is smaller, so again the pressure is going to be a little bit higher because there is more resistance to flow. All right. So, what about the glomerular capsule or Bowman capsule, as we called it? Right. Well, the layers surrounding the capillaries, as you can see in here, glomerulus capillaries, uh, they have these podocytes. These are called podocytes. Podo means feet, cyte means cells. These are the cells. You can see the nucleus, but it looks like they have little tiny feet, and the feet are coming in really next to one another. So what I want you guys to do, put your fingers next to one another, all right? And so if this is your fingers, for example, all right, the other hand, you could go right in it as well, like that, all right? And now you have tinier space to pass through, okay? Now pour it under something, I don't know, maybe water and some stuff in the water. Let's say something that doesn't dissolve. You could think of some sand, for example, all right? So sand goes inside of the water and then you pour it inside your hand as you put in your fingers next to it. What happens? A lot of water is going to go right through your fingers. The water is going to go right through the fingers. All right? You're not going to be able to hold much of the water, all right? Because these fingers are not really, really tight and sealed completely. There's a little bit of a space in between them, as you can see, all right, which resembles these filtration slits. What about the sand? Well, the sands are too large, so you could see, like, the grains of sand or rice or whatever you have in that water, it stays in your hand, all right? So, this is kind of like the stuff that does not pass. So the smaller the stuff, it passes this filtration slit and end up in here, in this space, which is called Bowman's capsular space or glomerular capsular space. All right. So this is called filtration. Filtration is based on the size. A majority of the things that are too large to pass, they can't. They stay within the blood. And then the stuff that are small, ions, water, things like that, they are going into the filtrate now, and they are going towards the proximal convoluted tubule, PCT, that we discussed. All right, so here is showing you the fenestration from the glomerulus. You could see the basement membrane in between, and you could see the podocytes and the filtration slit. So substances, they're coming in, and they're going in only if they are permeable and they are small enough. So that's just what happens uh, early on, and we are done with the first process, which is called the filtration. All right, so the filtration is done. The next part I would like to go over, it happens to be the PCT, proximal convoluted tubule. Now, what we're going to do is we're going to put our concentration of what is happening right over here. Proximal convoluted tubule is not really a straight tube, they're really convoluted. And I would say a lot of important stuff happening in here. Major reclaiming or reabsorption takes place in here. All right. So process B, I'm going to call it reabsorption. And we're going to see what is happening in there. So let's get to it. We're going to dive in side of the proximal convoluted tubule and understand what is going on uh, and try to make a sense of it. So I'm going to take this one and show you maybe on the next slide, all right, what is happening on the PCT. All right, so if we have uh, this convoluted area, all right, this is a portion of the PCT. And interestingly enough, one side, these are the cells. So think about as they have microvilli on this side. And same thing with here, they have microvilli. So think about this as a tube. So literally, it's actually, I'm cutting the tube, but I don't know if you can see it. It's supposed to be a tube, all right? But when you cut it in half and you're looking at it, it will look something like that, okay? So first of all, we need to have some kind of a reference point. So these are uh, microvilli, we know that. These are important for absorption because they increase the surface area, all right? Um, Sometimes this part is called as uh, apical end, all right? And this part right over here sometimes is known as basal lateral end, all right? These cells are unique. These cells contains a lot of mitochondria in them. So these are mitochondria, and I'm just showing you in one cell, but they're actually literally in all these cells. So lots of mitochondria. Mitochondria give energy to the cell, so that's got to be important. Why do we need a lot of energy in the cells, which we will discuss shortly. On the basolateral end, on the other side, uh, there are peritubular capillaries. So right over here, you have peritubular capillaries. Earlier, you notice these are the peritubular capillaries, see, right here. So that's in the vicinity, is around them. Okay, so one of the things I'm going to discuss and go one by one is water, obviously. So when the water is coming, remember water is capable of passing, all right? So water can go into two different ways on the other side, okay? Now, you might say why the water is going, we're gonna cover that. But there are important channels, all right? And sometimes these are as water channels. And let me just show you as a blue color. Pretend these channels are open, all right? These channels are open. This is kind of like a protein channel, and this is referred to as aqua pouring. Agua means like in Spanish, like water. Aqua pouring. Hmm? Aqua pouring is basically water channel, so you could put water channel too. It's fine. All right. So one way that the water is gonna go to reclaim them, because okay, wait a minute. Um, why do we need to reabsorb the water? Because if you don't absorb the water, you're gonna lose it in the your urine so much you're going to get dehydrated. That's not what you want to do. Remember, earlier we mentioned uh, that the excess material is supposed to come out, but if there is no reabsorption, all the water from the artery is going to end up in the urine, and your urine will constantly have a lot of water in it. Guess what? Now you're going to get dehydrated. You don't want to lose the water if you need it. Remember, our plasma, majority of it, 55%, majority of that 55% plasma was water. You have water inside of our cell, you have water outside of the cell, interstitial cells, or fluid. All of those require water, though it doesn't make sense to just like get rid of the water that is so precious for us. So we need to reclaim that. And that's what happens. The water is going to go back in here and eventually is going to end up right inside here. Now, how? Give me one moment and I will talk to you about. Now, these ones that, by the way, there are other cells as well that doing this. So I'm just putting one channel. It could be more than one channel. All right, there could be many of these on the cell, but these that they're using to go to the other side, all right, they're going right through the cell. So water is going to go right through the cell and end up in the peritubular capillary. And yes, you end up with, again, you could build up your pressure because your volume it did not change that much, and you're absorbing the water that you need. These are called um, these are called a transcellular pathway. Trans means within the cell. Transcellular. So this is an example of transcellular pathway. All right. Um, but the water can go other way to the other areas on the other side as well from the filtrate. It can actually go between the cells. So these cells, even though there are tight junction, but a tight junction, it doesn't mean that the water cannot go in between them. Tight junction holding these cells together, all right, it cannot transport substances from one cell to another, but we're not doing that. We are going in between, all right? So water can actually go in between these cells, all right? Maybe I should have used a different picture. So the water is going to go right between the cells on the other side. Can the water goes in this way too? Yes, because there is peritubular capillaries on this side as well. So yes, if the water goes in between the cells, this is known as paracellular pathway or paracellular route. All right, para means next to, cellular cell. Okay, so they're going right between them, right? Okay, now a couple of things that is worth note. One is that various ions are gonna go along with it as well. Remember, water is a polar substances, so uh, everything that you learn it can be useful here, and a slightly negative, a slightly positive. Okay. So because the water is polar substances, as it's going around, guess what? There are some ions that can go along with it. So for example, chloride can go along with it. These are ions, right? Calcium can go along with it on the part that is slightly negative. Potassium can go along with it. So you can see there are a lot of ions that are going with the water between the cells. Now, why not going into the water channel? Because they're not big enough. The water channels, aquaporin that we mentioned, is just for water. Ions, they cannot go in between because they just don't fit and it's not permeable to them. All right. But in this case, they are, so they can go. This is term is called solvent drag. So the water, as is going between the cells, is dragging this uh, solute with it. So solvent, all right, drag is dragging the various ions with it. All right. So and some urea as well. Now, if something is lipid, lipid soluble. We know, for example, certain hormones are like that and so on. They don't have any problem passing through. So they could just like go in right there. And you're going from high concentration of here, for example, to a lower concentration in here. So they could just go right through the membrane. Again, we discussed the process of diffusion. All right. Uh, let's go ahead and take a look at what else is happening in here because it's awful lot is happening. Okay, so I'm going to erase this because I want to use the proximal convoluted tubule again. So for that, I'm going to go ahead and erase this and create a little bit more room so that way we can concentrate on the other things. So two pathway, paracellular pathway, and transcellular pathway, the differences and what are being transported. Now, the major idea, why is the water is going to the other side? Uh, because the other side has higher solute. Outside of the tube always have higher solute than inside of the tube. We can look at that. But for now, I'm just going to erase a little bit of these so that way we can go ahead and take a look at other things. Let's erase this one too because we're done with it. So those are all in place. All right. But what we wanted to do is take a look at what is happening on the other side. Remember earlier I told you that there are mitochondria in here. So on the basolateral end of these cells, you have a lot of these sodium potassium pumps. I'm only putting one, it doesn't mean you only have one, okay? So there is a lot of sodium potassium pump. And again, you should know sodium potassium pump pumps 3 sodium out and bring in 2 potassium in. All right, and 3 sodium out, 2 potassium in. So what does that do? It will lower the concentration of sodium in here and it will increase the concentration of sodium up here. And guess what? There is a potassium leak channels in here. So the potassium, once they get filled or once they come in, they can also exit in here as well. Bottom line, a lot of potassium, a lot of sodium on this side, which are solutes. So you could put in a lot of dots in here, okay? And again, these solute are in the peritubular capillaries because the concentration is higher, and this created higher solutes. Now you're beginning to understand, I hope, why water is going to get reabsorbed. either between the cells or through the water channels we discussed earlier, all right, is because other side have higher amount of solute. Water always goes to the area with the higher amount of solute. And in fact, don't memorize this, but all the water reabsorption in the nephron are all osmosis of some type. All right, they're all osmosis. So there you have it. That is how and why the water gets absorbed. To give you an idea, water can get reabsorbed uh, in the PCT up to about 65% of the filtrate water can get reabsorbed in here, right? 65% of water. Yeah. That means majority. Having the microvilli, having the sodium potassium pump, having a higher amount of solute, and a lot of water is coming in at the first part of the filtrate, so that is what is happening in here. All right. So now we can go ahead and discuss other things that gets reabsorbed because we are talking about the process of reabsorption. So let me go ahead and erase a few things in here again. We already know the water gets reabsorbed, but there are other channels in here that are important too. So one of them being glucose channel, and as you know, the glucose is higher in here because the glucose is passing. On this side right over here, you have a lower glucose and so on. All right. So what do we want it to do? We wanted to reabsorb the glucose. Glucose are important for, perhaps we already discussed this, for our fuel purposes, for, energy purposes. Glycolysis is something that we discuss and is important to give, you know, uh, so many ATPs, 34, 36 ATPs and such. So it is rather important to think about that. All right. So what do we need to do? We need to reabsorb the glucose. Now, this is a secondary active transport. So if you don't know what that is, go back to the reviewing of the notes on chapter 3. All right. So because there is a lower amount of sodium and because the sodium potassium pump is involved, you're pumping the sodium that causes lower sodium inside. All right. Now, there is a higher sodium in here. The sodium is in here is higher. All right. They are positively charged. They cannot pass the membrane, but if they have their own, and this was a co-transport, uh, it's act like a facilitated diffusion, but it's actually bringing two molecules in here. And the driving force of it is this sodium potassium pump. So this is an active, secondary active transport. Glucose along with the sodium comes in. Obviously, the sodium that came in is going to go out again using the sodium potassium pump. And the glucose that came in is going to go through their channel and again end up in the capillaries. So you just absorb the glucose. Amino acid reabsorption is a very similar phenomenon. I'm just going to use a different color for that. So let's just use the red color in this case. All right. So same thing, you have amino acid and the sodium. And again, because you have lower level of sodium inside, a secondary active transport, sodium is coming in, but along the way, amino acids are coming away as well. So we know what happens to the sodium once they get in. They're going to go outside, and the amino acids, they're going to go through their channel as well. So again, you are reabsorbing amino acids. So a lot of reabsorption, as you can imagine, what is happening in here. All right. So what did we reabsorb? Well, we reabsorb sodium right here because some of the sodium ending up in here and ending up in the blood. We reabsorb potassium right here. We reabsorb glucose right here. We reabsorb amino acids right here. What else is there? Well, let's take a look. All right. So ions, that's what we discussed. Now for the calcium reabsorption, you need the parathyroid hormone, PTH. All right. Glucose we did, amino acid we did, water we did. Yeah, we're almost done. We have to reabsorb the bicarbonate now. Why do we need to use a reabsorb bicarbonate? Remember, bicarbonate is a buffer, so they play important role in acid and base balance. You really want to not to have that? Then if you take in something acidic, your acid is going to go up right away and cause acidosis. All right. So buffer is helpful. We don't want to get it out in our urine. All right. So we want it to be able to keep that in the blood, but they're small enough to pass, and they're negatively charged, so it's hard to absorb them right through in here. All right, in the PCT. So I'm going to erase this again because I wanted to use this PCT, and you have that on the video, so you could pause it, take a picture, and look at it again. But right now, I wanted to go over perhaps reabsorption of HCO3 and talk all the things that is happening, so that way we are done with the PCT. So let me go ahead and erase this part. I erased too much, but that's okay. Okay, so now we wanted to find out how is the HCO3, which is our bicarbonate, uh, working, okay? And we can go ahead and take a look at those. All right. So HCO3 or bicarbonate happen to be a negative entity. So as you know, negative things cannot, or positive things because of their polarity, they cannot go in, obviously, unless there is another mechanism involved. All right. So earlier I told you that you're trying to get rid of the hydrogen ions in the blood. Why? Because you wanted to balance things as well. So this is part of what's known as a secretion process, but there are certain hydrogen pump in here that are ATP driven. So ATP is needed, and they can pump in the hydrogen that is coming. All right, this hydrogen can combine with the HCO3, and there are enzymes, carbonic anhydrates. So once they are combined with one another, we're getting our familiar uh, formula. We're making a carbonic acid. Carbonic acid can dissociate to carbon dioxide plus water. All right, and remember, carbonic anhydrase is an enzyme. The CA is like California, right? So just remember that. Maybe it will help you. Carbonic anhydrase. All right. So carbon dioxide is permeable now. This is polar. And water, we already discussed how water can go in between the cells or within the aquaporin and so on. But carbon dioxide, they could just go right in. All right. So carbon dioxide, and there's plenty of water inside of our cells. So again, plus water. All right. Because these cells also have the enzyme carbonic anhydrase, you're going to make again same thing, H2CO3. But what is happening? Why are you doing that? Because you're not able to do absorb this. It was negative. All right, you turn it into another molecule. Now it's absorbable. All right, so carbon dioxide comes in inside of the cell, plenty of water. You have carbonic anhydrase. Readily, you could make the uh, carbonic acid, and now the carbonic acid is going to give you hydrogen ion plus HCO3. So the only difference is these reactions are going backwards, all right? And this hydrogen ion, again, it can be pumped through this ATP hydrogen pump outside. And this HCO3, there are also special ways that you could get this. Uh, in other words, you have uh, HCO3 enters the peritubular capillaries by the symport uh, and via the antiport of chloride. So what happens in here is kind of like a chloride shift. Now watch. Uh, Let me use another. Let's use this color. Maybe. I don't know if you can see it. All right. So now HCO3 is going to go out, all right, and chloride is going to come back in. So the chloride is in here, is going to go back in here, all right, and HCO3 is going to go end up in the blood, HCO3. It's kind of like a chloride shift. You're losing one negative, but then you're gaining one negative is not going to affect the cell's electron neutrality. And that's what happens in one sense. Sodium and also sodium and also the HCO3. This is kind of like uh, if you recall, when substances are going against one another, was an antiport. Antiport. But HCO3 also can go via the symport with the sodium channel. So HCO3 can go out with along with the sodium. Earlier we discussed about this, and this is also known as the symport. Don't worry too much about that. The idea is we just absorb the HCO3. So one of the things that happens, we totally change this molecule to another molecule so we could absorb them because there's water everywhere, including inside of the cell. Again, you're doing this, so now HCO3 is inside of the cell, and using uh, the chloride shift and such, we could easily exchange that, and that's what happens. Now, what happens to some of the carbon dioxide? Some of the carbon dioxide, they could, because they're permeable, they could end up in the blood as well. So no big deal. That's fine. All right. Some of the water can go through the water channel in there as well. So that's it. This is how the uh, reabsorption of substances taking place, if you will. And we just went over uh, pretty much all the reabsorption process in the PCT. The next step is now that we are familiar with what happens, at least for the PCT, all right. Remember the hydrogen ion, you can do the process C. This is called secretion. Secretion when you wanted to get rid of the things from the blood back into the tube. All right. So secretion process such as you know toxins, drugs, things that are still unwanted can happen in the this particular tool as well, and this is called tubular secretion, uh, and this related to the perhaps the things that you don't want to, you gotta get rid of them. All right. Um, Now, in terms of in terms of what if, before we go on to the next phase, we need to specify the secretion right here is hydrogen ion. There could be some drugs, all right, and NH4 plus, right, some nitrogen waste as well, all right. So those are the secretion. Um, Now, what if, and maybe you could think about that when you're doing the diabetes or thinking about the concept of diabetes and such. Uh, let's go back to the PCT. Okay. Um, let's erase this one more time. You see, uh, the amount that you could reabsorb is limited, right? You can't just like, reabsorb everything. Why not? Because substances, they're not sitting in here until they get reabsorbed. And that's important concept actually, all right? So when you have whatever, sodium or water or whatever, all those part of the filtrate, they're not just going to sit here. Remember the hydrostatic pressure, which is the heart contraction, there is a force in here that these are going. So as they're going, you're trying to reabsorb material, you know, throughout. So that is a big important difference in here. They're not just sitting ducks, right? Think about that I don't know, there's like an area or a river and the fish are going. So you have your fishing rod right there. You may be able to catch some fishes, but a lot of them are just leaving, right? So there's got to be other mechanism involved. I mean, water, we just mentioned 65%, we're getting it, right? So there's a lot of them are passing through. So that is actually an important concept. Now, the fact that if individual is diabetic, they would have a lot of these glucose, all right? They have glucose because of lack of insulin or whether it's a receptor or whether it's a problem with the insulin secretion, doesn't matter. The bottom line is your urine, your filtrate is going to have a lot of glucose. And we already discussed how glucose is going to be absorbed. Have to go through the same channel as the sodium. So glucose is going to go in through these channels along with sodium, all right? But there's way too many of them. They're not going to wait around to get absorbed. A lot of them, they're just going to pass through and end up in the urine. So the urine, if you kind of like, analyze it, you will find out that they contain a lot of sugar in the urine. And that explains why a person, for example, is, always thirsty, why a person is have a high blood pressure, why a person is keep drinking water, all right? Because the glucose now, the solute, you're gonna have a lot of solute in here now, all right? So not all the water is getting reabsorbed in here because of the amount of solute in here is more than before and so on. So you're losing a lot of water with it, okay? And that's why the person is going to the restroom more often and so on. So it's important to take a look at the details and understand the material as well. All right, let's go ahead and take a look at the next event, which happened to be right over here. And this is called a descending loop of Henle. All you really need to know is the descending loop of Henle is permeable to water. So again, water can get reabsorbed and go back into this peritubular capillary. Approximately process B happens in here too, and only water reabsorption, and that is equivalent to about 15% water gets reabsorbed in here. What's the physiology behind it? The physiology behind this is very simple, actually. Um, Outside of the tube, so let's say that this is our tube, and outside of the tube has higher osmolarity compared to the inside of the tube. So if over here, approximately 200 milliosmol, so if in here is, let's say, 300 milliosmol, I'm sorry, 500 milliosmol, inside would be 300 milliosmol. If outside is 700 milliosmol, inside would be, 200 less, 500 milliosmol, and so on. So 900, this would be 700, and this one would be 1200, right? This one would be 900, and this one would be 1400, and this one would be the 1200, right? So this would be 1,200. So you can see earlier I told you as you're going down, osmolarity increases, which this tells you osmolarity increases. Even inside of the tube, osmolarity increases. The bottom loop of the Henle has the highest concentration inside of the nephron, has a higher solute. Now, uh, why is the outside has higher concentration compared to the inside? We're going to get to that. But because if it's so, then the water as it's going in, they're getting absorbed. All right. Where are they getting absorbed? Right into the peritubular capillary. All right. So when they are getting absorbed, guess what? Now the concentration of the solute increases because you just reabsorb the water back in here. All right. So this is the capillary, all right? So these numbers are outside of the capillary. So then what happens then? Again, same thing. More water gets reabsorbed, concentration gets higher. More water gets reabsorbed, concentration gets higher. More water gets reabsorbed, concentration gets higher. So these are permeable to water, and this is classical example of osmosis. Osmosis, water goes always to the area that is higher concentrated, that is in here outside, and that's where the water is going inside of the capillaries. So that's it. That's the descending loop of Henle. So earlier we had 65% of the water got reabsorbed. In here we got 15% of the water get reabsorbed. We already up to 80% of the water total got reabsorbed. All right, we're going into the ascending loop of Henle now. The ascending loop of Henle, all right, uh, is this part. Ascending loop of Henle is a little bit different because of the fact that they are glycoprotein inside these cells. They have this glycoprotein in there, which cause it impermeable to water. So water again wanted to go outside because outside of the tube has a higher osmolarity. but they can't. Water cannot go through this because of this glycoprotein. And this glycoprotein prevent the water from going in. So water want to go out, but they can't. All right. So the ascending loop of Henle, 0% of water reabsorption takes place. All right. Nothing. All right. So ascending loop of Henle is impermeable to water. That's the important uh, thing that you wanted to get at. However, the ascending loop of Henle, all right, uh, there are important sodium, potassium, and chloride ion pump. So basically, what you need to know is that they are permeable to NaCl, all right? So salt. NaCl are permeable in there. They are a special pump. Again, these pumps can is called sodium, potassium, and chloride pump. So two chloride ions goes out, and so as the sodium and the potassium comes in. So basically, what happens is at the end results, just take my word for it, is sodium chloride is permeable. All right. So because of those pumps. Now you're going to begin to see why outside of these tubes were higher, because this is solute. So that's the reason why outside is higher. Okay, that's the reason why outside is higher concentration compared to the inside. But if you take away the sodium chloride or the salt, which is a solute in this case, what do? The numbers is going to go opposite. So in other words, now you're at 1200, now you might be at 1000. More sodium chloride is going to get reabsorbed and go out. Not all of them gets reabsorbed, but what happened is, again, the number goes down, 700. And again, more sodium chloride. So you could see as sodium chloride goes out to make the higher solute concentration outside, these numbers is reducing, all right, which setting a stage for the next part, which happened to be the distal convoluted tubule. So the bottom line is that this part right over here, reabsorption of sodium chloride taking place. So still is a process B is happening, but none of the water gets reabsorbed in here due to the glycoprotein that we discussed. All right. Um, so we can go ahead and take a look at the last portion, and that is, this part right over here, distal convoluted tubule and collecting duct, which they operate via the hormonal mechanism. This hormonal mechanism is essential and is important, and we can discuss this, see if we can make a sense of this hormonal regulation, perhaps. So we can discuss these two together, and we can see what is happening on the collecting duct as well as the distal convoluted tubule. All right. So remember, some of the DCT right over here, you have a calcium reabsorption, and you need parathyroid hormone for that. Parathyroid hormone, um, you know, is essential. We discussed parathyroid hormone earlier, a similar concept, but that's what is happening in terms of the uptake. These are channels in the apical membrane, and is a passive process. The parathyroid hormone helps these channels, and you are absorbing the calcium. Okay. Now let's go to the DCT and collecting duct for that matter. For now, I'm just going to put this as remember we are here, all right? And now we can go ahead and take a look at distal convoluted tubule and the collecting duct. All right. Now, uh, distal convoluted tubule and collecting duct. What I wanted to emphasize is that there are under hormonal regulation. Okay, they are under hormonal regulation. What does that mean? Well, let's first begin to talk a little bit about different glands that are involved, and then we can dissect this a little bit further. Okay. Now, uh, on top of the kidney, so this is the kidney. On top of the kidney, there lies adrenal gland. Adrenal gland releases a hormone known as aldosterone. All right. And in the brain, there is an area is known as anterior and posterior pituitary. Right now, we don't care about the anterior. We're just looking at the posterior pituitary. Posterior pituitary produce they release the hormone ADH, anti-diuretic hormone. This is also known as vasopressin, all right? Anti-diuretic hormone or vasopressin, okay? So these two hormones are playing an important role in our thirst and balancing the water, and they play an important role in the regulation as well. So we're going to get to all of that hopefully soon, but for the time being, I just wanted to tell you guys what they do. All right. So um, let's see. As far as the ADH goes, so let's make these cells, right? These are the cells of the collecting duct and the, uh, you know, uh, ECT. So the filtrate is coming. Remember, 80% of water already got reabsorbed. So, and earlier I showed you that you took out some of the solute, so the filtrate has less solute. You're on top in the cortical areas right now, and you're beginning to go towards the medulla. All right. So the filtrate is coming. ADH. Now there are some V2 receptors in here that they can take in these ADH. Now remember the ADH, there are hormones, so they travel via the blood. I guess we can put the blood in here. All right. And eventually with the blood, they are being transported from the glands. And I gave you ideas about it in the previous lecture. So now they can bind to the receptors. All right. Binding these hormones to the receptors is basically involve a G-link protein that you already know, and this G-link protein is going to cause the DNA and RNA and translation process to take place, and so on. And as a result of that, what happens is you're manufacturing a new type of a protein, and that new type of a protein is basically aquaporin. So we're making these proteins known as aquaporins. Now, normally these cells have plenty of these water channels, all right? But now you're having more water channels. All right, so if you have more water channels, whatever the filtrate is, remember now it contains 20% water, all right? So some water now, they could go right on the outside of the tube. And as you know, the outside of the tube has higher concentration of solute, as I mentioned to you. So this side has a higher solute to begin with, and the water ends up in here. Now, you're not going to be able to absorb or reabsorb 20% of the water that is remaining because your urine consists of some water itself as well. So this depends on your need. If you are dehydrated, you'll be absorbing more. If you are not dehydrated and you're hydrated, you'll be absorbing less. So in a way, it does is fine tuning. All right, so ADH, the more ADH you will have, the more of these water channels or aquaporin, and the more absorption of the water you're going to get. So you can maybe not memorize, but look at it. Uh, when you are dehydrated, during dehydration, your brain that has a thirst center, you know, you get activated. There's a lot of signals going on. And finally, the pituitary gland, posterior pituitary, releasing ADH. More ADH is going to be released. And as a result of that, more water reabsorption. Uh, and that's what we want because we are dehydrated. We wanted to reabsorb the water better. And as a result of that, you're going to have lower urine output. All right. In other words, your urine is going to be very concentrated. That's what's going to happen. All right. So ADH is going to create more water channels and keep these water channels open. So that way you can go ahead and do that. Now, what if you already absorb all you want, or whenever you're hydrated, then there will be less of these ADH. If there are less of these ADH, then there will be less of these channels, as well as less reabsorption of the water. So now, instead of, let's say, 18% of water getting reabsorbed when you were very dehydrated, now you're very hydrated. Now you might actually, do just a little bit, 5%. So what happens then during hydration? Opposite happens. During hydration, you have lower ADH, therefore you have less water reabsorption. And as a result of that, you will have a higher urine output. And you know that already. If you keep drinking water, you need to use the restroom more often, okay? Because your urine output is getting higher, all right? And the ADH is lower. Now, this hormonal control would take a little bit of time. It doesn't happen like right away, okay? All right, so that is pretty much what is happening in terms of the ADH. What about the aldosterone? Aldosterone, it belongs to a family of the steroid hormones. So basically, they could go right in. They don't need these receptors. So, I don't know if you wanted to kind of like maybe show as a circle. The other one was a triangle. All right, so these, they can go into the blood. And then again, they can go right in and cause the nucleus to, or the genetic material to make the protein, and this protein happen to be sodium potassium pump. All right, so this sodium potassium pump 3 sodium out and bring in 2 potassium in. Some of the potassium is going to go back in here. Some of the potassium is going to end up in the filtrate. The bottom line is, when you have so many of these sodium potassium pump due to effect of aldosterone, guess what? They are pumping a lot of solute on this side. So there's going to be a lot of sodium in this side, a lot of potassium in this side. All right. So this is going to increase the solute. This is another reason why outside of the tube has higher osmolarity. All right. So now what? Well, if it's solute, when the water is coming, guess what? They could go paracellular or they could go transcellular. Either way, they can get there a lot more because the osmosis that we discussed is happening. Osmosis will be faster if you have a higher amount of solute, and that is exactly what is happening. So not only water gets reabsorbed in here, but also due to higher amount of solute, you have more concentration of solute on this side. So the driving force for the water or osmotic pressure is going to be higher in this case. So uh, effect of aldosterone, increase in sodium, potassium, pump numbers, so you have a lot more. Number two, again, these cells, they have a lot of mitochondria too, so they could feed off the sodium potassium pump, and uh, the pumps, uh, the pumps, they are working at a faster rate, all right? So what that means is they're pumping fast, there's a lot of them, and you're creating a lot of solute on this side. So the water, either they could go right through the aquaporin or between the cell, transcellular and paracellular, both of which they are doing it. So during dehydration, you would have higher amount of aldosterone, and therefore you would have more sodium potassium pump activity. Therefore, you would have more water reabsorption. And therefore, again, the same thing as this one. Therefore, you would have lower urine output. And during hydration, exactly opposite would occur. Exactly opposite would occur. All right, lower aldosterone, lower sodium, potassium activity and number. This one, activity and number. That means more of them. And therefore you would have lower water reabsorption and you would have higher output during the hydration. So basically it does exactly the same thing as this one does, all right? So guess what? ADH, higher ADH during the dehydration. Don't go memorize this, think about it. The same thing as a higher amount of aldosterone. So they work cooperatively with one another, which makes sense. Which makes sense because if you have higher amount of solute, guess what? Now you will have, more water channels too. So more opportunity for water to go towards the higher amount of solute, and basically it serves the purpose. All right. So now you should have a good idea about all these areas that we discussed. We looked at each area, what happens on first, what happens on the PCT, what happens in all these areas and so on. And hopefully by reviewing the material and so on, you learn a little bit about this phenomena as well. And we can carry on the rest at 10:35 or next time.